Hello, hello, hello. Okay. Okay, let's go. Okay. Got my charger in. Working out what I'm actually doing. Okay. Right. Oh God. Okay. Let's get down to some A-level biology. Okay, discuss the energy, well, it's too hot for a hat. Discuss the energy transfers that take place inside and between living organisms. So, checking my mic's working. Good, good, good. Okay. Right. So, energy transfers is an essay that's been predicted could come up this year in biology by quite a few different YouTube people. So, you could be asked to discuss the energy transfers that take place between organisms and ecosystems, or between different organisms, or inside organisms. And this is kind of like a made-up question that's kind of covering both. But be prepared that it might just be the flow of energy inside organisms, or the transfers inside organisms, or between organisms. So let's get into this. Okay, anyone want to put up on the chat what they think? When you're doing an essay, remember an essay is going to be 40 minutes, 45 minutes. This means they say you've got to talk about 4 topics, possibly 5. That way, if you screw up one topic, you've got 4 others to gain the maximum marks. However, you have to work out in your head, if you're spending 40 minutes and say you're doing 5 minutes planning, then that, if you're going to write 5 topics, it leaves you 7 minutes per topic. If you're going to do 4 topics, then you've got a bit more time. You've got like 9 minutes or something per topic, 9-10 minutes per topic. So you've got to be, you do need to think about how much effort you put into a 5 marker at the end of paper 1, one of the content knowledge ones at the end of paper 1. And if you think about how much effort you put into that, well, you're going to need to, you're going to be spending more time on each one of these sections than a 5 marker. So you really need to not do it down at GCSE level. It needs to contain A-level key terms. You can't just breeze through. You've got to really hit the details, and then you link it back to the question, obviously. So it could be discuss the importance of it. Most of these are to talk about the importance, which means you just say what would happen if it didn't exist. And you link that back at the end of saying all the detailed information. But yeah, just think about how much effort you need to go into each of those 4 paragraphs or 5 sections, or however you want to call it. So what do people think would be the 4 sections or 5 topics or subtopics that we could talk about with energy transfers, say inside living organisms, or between living organisms, or between living organisms and the environment? So we can just do a mixture of all and see if we can come up with some energy transfers. So feel free to put on the chat if you have an idea of a way in which energy can be transferred. So the more difficult ones are the ones inside living organisms, and you're looking at really, I'm just checking if anyone's it's not I'm really around tonight, don't blame them. So I would say one of the biggest ones you're looking at is photosynthesis. And of course, when you're thinking about energy transfer, between the environment and organisms. You've got sunlight, which is a type of energy, and it's coming down and being turned into chemical energy in the leaves of plants, obviously in the chloroplasts. So we've got photosynthesis. So this should be one of your big four when you're talking with energy transfers, regardless of whether it's inside an organism, which we'll be talking about inside the chloroplast, or whether you're talking about transfer between organisms. Photosynthesis is going to be one of your key four topics. And here are the details that you're going to need to discuss. So I can actually get rid of that. So the details you're going to be looking to discuss with photosynthesis, you've got, obviously I'm not going to write a full essay out, that's just too long. So you're looking at really the light dependent reaction, discuss photoionization of chlorophyll. So you're looking at chlorophyll A, chlorophyll B, what role do they play in absorbing the light energy? What wavelength of light is absorbed? Do you remember what wavelength it is in nanometers? Most of the light is actually reflected off of the leaf, and only a small percentage of sunlight enters the leaf. Do you know your percentages on that? Then, so you might want to check that out. And then you also might want to discuss how the chloroplast, how its structure is adapted for the job of absorbing sunlight. So you can talk about the membrane, the stroma. Then you want to talk about mainly focus on the light dependent reaction, because what's happening in photosynthesis in terms of energy transfer is that energy is being transferred into electrons. Well, it's not becoming electrons, but the electrons are gaining that sunlight energy, or a small portion of it. Most of it's reflected or not absorbed, or doesn't even hit plants at all. But a small portion of that energy that goes into the chlorophyll is exciting electrons, and the energy from the sun is now in the electrons. And so you need to talk about the electron transport chain, and then how some of that energy is transferred to do active transport of hydrogen ions to set up a concentration gradient for chemiosmosis. Now, the end result of that is the hydrogen ions going through ATP synthase and the energy getting locked into the bonds ADP and PI, inorganic phosphate, to become ATP. So now that's a small percentage of that sun energy has ended up in a molecule of ATP. Okay, so there's our energy transfer from light to chemical. But you need to explain those steps. So make sure you know your light dependent reaction if you're going to focus a whole paragraph on this, and you should be able to spit out all of that knowledge in like 5 or 6 minutes. Now, some of the energy goes on, or some of those hydrogen ions that were actively transported using some of the sunlight energy. So we always want to keep referring to energy and linking it back. If you're just talking about hydrogen ions and chemiosmosis, but you're not linking it back to energy transfer and the fact that that energy came from the sun, then you're not really stressing the links back to the question. So some of it goes to reduce NADP. Now, they both go on ATP and NADP reduced to the light independent reaction. You don't need to talk about the whole of the Calvin cycle or the dark photosynthesis reaction or the light independent reaction. You just need to talk about the part where ATP and NADP reduced are involved. And that, of course, is changing GP into TP. And then that goes into carbohydrates, and we get our energy basically converted into carbohydrates. So that's really what you're going to talk about there. So we started off with sunlight energy, how it gets into the chloroplast, how they're adapted to do that, the wavelength of light, the photosynthetic pigments and accessory pigments and stuff that are involved in that, the excitation of the electrons, which are then carrying a small part of that energy, their movement down the electron transport chain, the energy used for active transport, the hydrogen ions being moved across the membrane, then coming back through ATP synthase by chemiosmosis, the energy ending up in the ATP from ADP plus PI, and then the reduced NADP, and then that energy being transferred in the conversion of GP to carbohydrates in whatever steps you want to go there. So that's what you need to talk about for one of them. Now, can you do that in 7 or 8 minutes? You should be able to, because you've been revising for your paper 2. You'd have revised photosynthesis really well. So that's a key one. Now, any questions about that, feel free to put up on the chat if you have any questions about that. Okay, let's look at the next part of that. So I'm going to just minimize that. So I would definitely, if you're given energy transfers, that's the biggie, sunlight into plants. So that's where you want to be starting. I would also take 5 minutes out of your 45 minutes for the essay, or your 40 minutes for the essay, depending on how much you need. I would also take 5 minutes out to plan. And with a plan, you can literally just come up with the topics you're going to talk about for this. Remember, you can't just talk briefly in GCSE language. If the same topic at A level appears in GCSE and you don't use any key terms from A level, so if you're just talking about diffusion in one of these essays and you're just using GCSE terms and nothing funky from A level about something specific to A level, then you're not going to get the mark. So yes, diffusion occurs in A level, but you need to talk about all the funky stuff. So respiration, we've got energy transfers going on again. So you've revised for paper 2. You should have a pretty good idea of how to say 5 marks or 5 key points about respiration and how the energy is transferred. Again, similar to photosynthesis, we've got energy transfer there. This is again within organisms. Another good place to talk about energy transfer, whatever that is. Yeah, another good place to talk about energy transfer is in muscle contraction, or actually, I probably prefer nerve transmission than muscle contraction, but you can do muscle contraction as well. You've got energy being transferred there, ending up as kinetic energy when you actually get the power stroke and your muscles move. And again, that's all coming from ATP. And we can talk about the energy locked in ATP that you need in order to reset the cross bridges and all of that kind of stuff. But nerve transmission, again, is a good one, because with nerve transmission, or the formation of action potentials, you can talk about the receptor and how the energy is transferred into electrical impulses in the receptor. So you've got like rods and cones in your eye that are going to basically take light energy again to chemical energy and then to electrical energy. And then you can talk about how the energy is transferred along the nervous system and that kind of stuff. You can talk about how it crosses the gap. How is that energy crossing the synaptic cleft? Again, GCSE knowledge would be if you just talk about movement across the synapse, diffusion of neurotransmitter across the synapse. That's GCSE. So you need to refer to it as the synaptic cleft, because that's A level. If you're just calling it the synapse, that's really GCSE. So you want to talk about your calcium ions, your movement of vesicles to the presynaptic membrane, and then the fusing with the membrane, and then the release of the neurotransmitter. And you want to explain the whole thing, but you're really reminding the examiner that you're talking about an energy transfer from electrical impulses, so the energy is as electrical, into chemical in the synaptic cleft, and then back to electrical, so the energy is being transferred across. And again, that's another one inside organisms. So if you're looking at energy transfers outside of organisms, we've got another paragraph you could talk about here. Again, you've got food chains and producers, blah, 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 blah. You've got biomass, consumers, dry mass. So you should know what GPP is and what MPP is and what R is. So you've got gross primary productivity, net primary productivity, or production, sorry. And you've got respiratory losses. So you need to be able to talk about that. Talk about primary productivity, which is the rate of energy transfer, and that's in kilojoules, hectares per year here. And then you've got the net production into consumers is only 10%. So you can calculate net production in consumers as N equals I minus F minus R. So you need to know all that. I is like the amount of biomass, or the amount of energy ingested in kilojoules, minus feces, F for feces, minus excretion like feces, urine, and minus respiration. So really, that should actually be F plus R in there. Just realised that. I was just banging this out well quick. So yeah, so you're really looking at net production in consumers. Net production in consumers which is 10% of the energy they get from of all the plant material and biomass they eat. They only get 10%. And the reason is 90% is lost as feces, urine, and respiration. So you want to be able to put that equation there. Now, if you do that as a rate per area, per time period, then we call it secondary productivity, the rate of secondary productivity. And that is for consumers. It's called secondary productivity. Your primary productivity is the rate of energy transfer in plants, which is really small, whatever that is, of the amount of energy that actually hits them. Of course, the plant loses about 50% of its energy from respiration. So you've got your net productivity, net primary production, going crazy, is your gross primary production minus your respiratory losses for plants. Okay, cool. So you want to sort of discuss all of that and keep referring back to the question about energy transfer. So we're basically, this limits the length of food chains and pyramids of biomass and shows us why they're in pyramid format. You can also bring in lots of other stuff. Anyone got any questions? Yeah, no point. The GCSE, I haven't really been doing it. My channel doesn't really, thank you team, my channel doesn't really focus, and Yoshi, on A level. It's mainly GCSEs. Thanks GDF, but like I'm only just starting to hit the start up the A level stuff and the paper 3 exam. So next year I'll be doing like loads of A level, and this summer I'm going to start A level for people going from transitioning from GCSEs to A level. But next year I'm going to be doing live streams for all of the A level exams, chemistry and biology. But yeah, this year we're just starting up and having my own students that I teach, and then doing live streams for the GCSEs. It was just too much to organise. Other sections, thank you Betty. Other sections you have are general points or all points within one paragraph. Okay, these are general points. So you would need to, Betty, you would need these sections. These are these are like 4 different sections you can talk about. But this is not like, this is going to get you a limited amount of marks if you put this. This is a plan more. This would get, you'd have to expand on all of those points. Yeah, there are beyond the specification points you can put into this. There's quite a lot actually beyond the specification. Obviously, the one I was talking about on my earlier live stream, which was like a 30-minute one I did earlier today, was the old cholera, not chlorella. Don't get it mixed up with the green protist that photosynthesizes, but cholera, the bacterium with the corkscrew flagellum. You can smash that into about 10 or 15 different essays, which I showed in my earlier live stream. This one is a little bit more specific, and there's a whole bunch you could go on this one. So I'll show you some of them. Obviously, you might want to research some of these yourself to add stuff in. And so this is a beyond the spec bit that could potentially score you the marks if you're talking about relationships between organisms. But it's quite limited. I've got some other ones I'll go through in a second. So you've got like, there's a plant called the bird nest orchid. It's actually got a Latin binomial name, which I can't remember. And there are certain plants out there which are non-photosynthetic. So you could discuss that. When you're doing energy transfers, you're always going to be talking about photosynthesis. So you could discuss, hey, there are some plants that can't do or don't do photosynthesis. They're non-photosynthetic, and they actually have a symbiotic relationship. They work together with fungi in something called mycoheterotrophy. Okay, now plants are normally autotrophic. Autotrophic means make your own food, essentially, make your own complex organic molecules, doing photosynthesis. But there are some plants, and there's this orchid. What it does, it lives on trees. It grows on the roots of trees, so it's like wrapped around the base of the tree, and it grows up. And it works with fungi that grow on it, and together they break down the nutrients at the base of the tree and absorb those nutrients. The fungi help them absorb the nutrients. So yeah, it's I never know how to say that, mycorrhizae fungus. Yeah, so that's how you spell it anyway, I believe. So it's called mycoheterotrophy. So that's a small part of something that's outside the spec. But what else could you come with energy transfers outside the spec? Let's have a look. Did I put anything else? Okay, yeah, I've got a couple of other things. And again, like I said, those points were only plan points. You would need to expand on those. So you'd need to expand. If I just go back one sec, with these points here, mycoheterotrophy, good question, Betty, is the description of the process by which that organism gets its energy. So we normally split the world into fungi, which are saprotrophs. They're saprotrophic, which means feed on dead material. Okay, they grow those hyphae through their mycelium into dead matter, like rotting leaves or whatever, like mushrooms, and they suck up the nutrients. They excrete extracellular enzymes outside of their body and then break down the complex organic molecules and suck the bits up. They're saprotrophs. All animals are normally described as heterotrophs, because we have to consume, we're consumers, so we have to consume things and digest them in our bodies. Plants are called autotrophs, and green algae and green plants are autotrophs because they photosynthesize, and cyanobacteria and some protists as well have chlorophyll in their cytoplasm, so they can photosynthesize, and they make organic molecules in their body. This is another type that's not in the book anywhere. It's called mycoheterotrophy. The myco means it involves fungus, because myco kind of is always to do with fungus, some Latin. And heterotrophy means kind of like eating as well, like consuming. Plants don't normally consume, but this bird's nest orchid, called a bird's nest because it looks like a bird's nest. You know, like a bird's nest is brown because it has no chlorophyll and it doesn't do photosynthesis. It looks like a bird's nest when it grows up the base of a tree. And this orchid works with the fungus in its roots to absorb the nutrients from the base of the tree. So that's just something, but that's only a little part. Now, what I was saying about these bits, if you were doing this essay, energy transfers, you will need to expand on photoionization and talk about accessory pigments, the wavelength, what is it, 400 nanometers to 700 nanometers, the red and the blue light mainly, that's absorbed by plants. You'll find that all in your CGP in your book. You should know that from your revision for paper 2, photosynthesis, and you should be able to discuss the electron transport chain and explain that the energy from the sun has gone into the electron. So you'd need to say all of that stuff that I was waffling on about earlier. These are just brief points, like, of what you would fatten up into a big chunk. But my main thing is you could spend 40 minutes writing practice essays. Now, AQA have asked 40 essays in the last 20 years. They ask 2 every year. So there's like, how much can you get done? It's better to do this and have that in your head and then rely on your adrenaline in the exam. Yeah, I'm going to be able to bang out a load of stuff about, if I just know energy transfers, I need to talk about photosynthesis as a separate topic, respiration, action potentials, and then like food chains and productivity within the ecosystem. So you could just do that. You could just focus on those 4. But you'd need to fatten those up and make them into solid bits of content. Right, beyond the spec, yeah, there's that little bit, but you might prefer there's this one. This is another bit. This is a bit longer. Feel free to put any questions in the chat. So this is a bit longer. This is... Again, this is just to save you guys, like, you can research these yourselves. Like, you literally can put into Google pelagic food chains, find out what the hell that's about. Pelagic food chains means food chains that start with single-celled organisms like phytoplankton. Phytoplankton are a type of plant that plankton are in the oceans. and they absorb sunlight. Now, generally, the problem when you're in the ocean, this is beyond the spec stuff, right? So the problem when you're in the ocean compared to land is sunlight, yeah, it's got to penetrate through the water. So unlike just hitting a tree on land, it's got to penetrate through the water and get down to these phytoplankton. So generally, you would think that food chains should be shorter. There's less energy. There's less energy to be transferred because they're absorbing less energy. The primary production is less, okay? So in pelagic food chains, these are food chains with single-celled organisms, mainly in the ocean. These oceanic or marine food chains, the primary production is less, understandably, because the nutrients, that the minerals and nutrients are going to be a lot further apart, and the sunlight, there's a greater chance of it being absorbed in the water, right, compared to on land. So there's even less sunlight getting absorbed by these single-celled photosynthetic organisms like phytoplankton. Now, so actually they say they have a 5 times lower primary production. Primary production, like how much of the sunlight is turned into biomass. So how many carbon molecules do they make? It's 5 times less. However, the food chains are actually longer in the ocean. So they actually normally go to 5 steps, where your land food chains normally stop with at 4. These can go to 5 trophic levels and even more. And the reason being is that because they're digesting single-celled organisms, the consumers, they're feeding on phytoplankton, they don't lose biomass through all that undigestible stuff. Like, you know how if on land a goat eats a tree, it might poo out or excrete a lot of undigestible tissues. Well, there are no tissues in a single-celled organism. It's just single cells. So it's like when you eat corn or something, when you eat, those kind of fungus protein burgers. So it's just single-celled mush. So they're going to absorb it really rapidly, and the speed of energy transfer through an oceanic food chain is a lot more rapid. Also, they are more metabolically efficient. Now, that is not the spelling of metabolically. That says metabolically. I don't know what I'm spelling there. So that's supposed to say metabolically. They're more metabolically efficient. And that's because organisms in the sea are known as ectotherms, keyword. You may have learned what an ectotherm and an endotherm is already in AQA biology. So an ectotherm, basically cold-blooded animal, they can't regulate their temperature. But because of that, ectotherms, they don't lose heat. Like right now, I'm losing so much heat. I can feel it coming out my armpits. I'm sweating. Because we regulate our temperature, we lose a lot of energy as heat. Those animals in the ocean don't lose as much. So the 2 reasons that food chains are longer, despite having a lower primary production, primary production in the sea is less, meaning the plants absorb less sunlight and turn less of it into organic molecules. The reason that the food chains are longer is because it's more metabolically efficient, because they're ectotherms. They tend to be more efficient with their energy transfer. They don't have to regulate their temperature. They don't regulate their temperature, so they don't lose any heat like that, basically. So that explains why they have an extra trophic level. So you could go on to that if you want to do transfer of energy, but that would mean remembering some of this for Wednesday, so whatever's easiest. I don't even know what's in this little box. This could be completely irrelevant. Now, this is in your... Those of you with the AQA book, is this in the AQA book? Oh no, this isn't in the AQA book. You've probably heard, this is another one, or another alternative. Now this is, I would pick one of these and stick with one. The chance that I know that several people have said energy transfers might come up, but it might not. So it's like you could waste a lot of energy doing that. That's why I would, excuse the pun, I would recommend only planning out essays at this stage and getting the general gist, and then relying on your subject knowledge to carry, and adrenaline and everything to carry you through. So plan out the 4 topics you're going to talk about, briefly keyword down, and try to do, like, you're better off knowing 15 essay plans, because then you've got a chance of minimum getting 15 to 20 marks. than knowing 3 essays really, really well and putting it all on. Hopefully someone's predicted the essay right. And if they haven't, you're sitting there going, oh, I could have done a 25 marker on that. You're better off covering a wider spread. Yeah, this is another one. This is actually from the International Baccalaureate. So those people who do sixth form IB, you have to know this. But yeah, very briefly, I'll just explain that. But again, whichever one's easier to remember for you. This is just talking. You could link this one into global warming. This is one for global warming, really. But yeah, you can read that if you like. That's just Greenland's covered in snow, but there's these black patches made of volcanic dust and sand and soot from pollution. So you can link that into combustion, global warming, and if carbon dioxide, incomplete combustion, releasing soot. So if you get something along those lines, you get these black patches on the ice and snow in Greenland. And every year when it snows, they get covered over and layers form. But they absorb more sunlight because they're black, and black absorbs radiation better. It absorbs infrared radiation better than white, which reflects radiation. And what's happening is these black patches are heating up, melting the snow, and revealing more of the black patches from the previous year that were covered in snow. And then the black patches, the surface area of them is getting bigger and more sunlight's being absorbed. And this is causing whole ice sheets in Greenland to melt. And you've got cold water going into the sea, and this is going to affect the distribution of organisms. It could cause flooding. There's all sorts of things. It's going to destroy habitats as the ice sheets melt and all of that. So you could kind of talk about that. You can link that to energy because it's absorbing energy from the sun. So we've got energy transferring into being absorbed by these black patches, if you want. But you could link this into positive feedback, because a mechanism where something happens, and the more it happens, it just further increases the level. That's positive feedback. It's not returning to the original level. As more heat gets absorbed, more snow melts, more black patches are revealed. They've got a name as well. It's called criconite. I don't know if it's written there. Yeah, criconite is the name. These black patches. The more of the criconite is revealed, the more it absorbs, and it's positive feedback. So you could get something called feedback loops that could tie in there as well. Yeah, so the pelagic could go, yeah, so the pelagic one, you could bust the pelagic one on any spec point to do with energy and ecosystems. Yeah, you could work that into anything to do with energy and ecosystems. So that could go into anything, that would be a beyond the specification to do with energy and ecosystems, yeah. Or this, so yeah. Or you could go back and do this one here, which is, again, you could work that into any point, really. Your paragraphs don't have to link, but they all have to relate to the question. I just would like those, you just got to keep relating them to the question. Either, if it flows naturally, you just flow it off as a separate paragraph. They're not going to downgrade you for either of those. They won't downgrade you for either of those at all. They're not going to downgrade you at all for this whole, oh, write 4 or 5 paragraphs has only come about because on the mark scheme, it said, you've got to cover 4 topics. 6 years ago, you had to cover 5 topics. Now they've reduced it to 4, but that doesn't make it easier. But they say you've got to cover at least 4 subtopics, and they've got to be subtopics. They don't have to come from completely different parts of the spec. For instance, photosynthesis and respiration together in the kind of same unit, they count as 2 different topics. So yeah, just make sure it's relevant. You don't start talking about stuff that's not relevant. So when I was talking about photosynthesis, I was talking about where the energy goes. So the energy is in the electrons that are moving. I start talking about, I suppose photolysis involves energy, because the energy from the sun splits the water molecule into oxygen and hydrogen ions. So photolysis can be mentioned there, but there's things which you might not want to mention because they're not directly related to energy, basically. Also with energy transfer, something I didn't mention, I didn't really, but it was under the respiration. Oh, I won't flick back. It was under the respiration, but you've got the whole ADP plus PI makes ATP. That's a huge thing to talk about, and there's lots to talk about there related to energy transfer. So, got another one here. Oy. How do bacteria affect living organisms? So this is another essay. So you could come up with 4 points on this. What would be your 4 points on how bacteria affect living organisms? Microbes, bacteria, all of them could come up. So it's one that hasn't come up for a while, but in the past they've asked like twice before, like how microbes affect living organisms or bacteria. So yeah, cholera would be the beyond the spec. Cholera works for so many things because it works on the cell membrane. So if membranes come up, I know some YouTubers are saying they've got a hunch membranes are coming up. Well, cholera really works on that because the receptors that the toxic protein it makes bind to are found on the cell membrane, and yeah, they're carbohydrate receptors that it binds to, and they're only found uniquely on the epithelial cell membrane. That's why cholera only affects the small intestines. Yeah, so I would go for cholera for beyond the spec. And you'll actually find, I think it might even be in the, you might have cholera in the book. It's also in the old spec book as well, cholera, so it's a good one for that. What about for, so think about where the bacteria appear in your CGP book or in your A-level book, wherever the bacteria appear. So what sort of topics? And this is how I would prepare for the essay. So if I'm going to waste an hour of my time in the next 2 days, yep, brilliant, nitrogen cycle. So if I'm planning the essay, I'm going to write nitrogen cycle. Now I'm going to say to myself, what do I want to talk about? What do I want to know about the nitrogen cycle? I'm going to rely on my brains to just pull out the information I've done for my paper 2 revision. Yeah, immunity, great. Great, immunity. Now, immunity, we can go down all sorts of paths. We can do phagocytosis. We can talk about vaccines, attenuated pathogens, and all of that. Formations of APC. So you've got a nice fat paragraph to talk about there in immunity. Obviously, in the nitrogen cycle, it would really help if you can remember your nitrobacter, your azobacter, your whatever they are, rhizium, whatever, rhizobium, all of that. If you can remember the names and what each one does. If you can't, don't worry, they're not going to punish you for like one mistake or like, but irrelevant stuff they will punish you. So don't waffle on about the nitrogen cycle and how lightning fixes nitrogen into the soil if it's asking you about bacteria's involvement. You've got to keep referring to how bacteria are involved. And a lot of these essays say the importance. So you want to say, well, it's a really important role that the bacteria play, because if we didn't turn ammonia into nitrates, plants wouldn't have nitrates that they could absorb into their roots and use for growth, okay, for making amino acids and proteins, etc. So you can stress to the importance. You don't have to go too much off on a tangent, but it's good to keep reminding the examiner, telling the examiner, look, you might think I'm just waffling here, but I'm going to show you why it's important. But I keep telling you it's important because of this. So often they say, what's the importance? So then you want to use the word important. You want to say bacteria are important in the nitrogen cycle because nitrobacter do this. And if they didn't do this, we couldn't get this. So kind of want to go through that. Yeah. So you've got nitrogen cycle and immunity. You're going to need 2 more to do with bacteria. So what about in your, you've got obviously genetic engineering. So hopefully you all did your revision for paper 2. You know a little bit about your in vivo cloning or whatever, or your stuff about bacteria. plasmids, using plasmids from bacteria in technology. We use them as vectors. Remember, some of that is GCSE, just saying, oh yeah, bacteria have a plasmid. Don't think you're scoring brilliant A-level marks by pointing that out to the examiner. Even restriction enzymes, you know, you need to say endonuclease and you need to say ligase, and sticky ends, complementary sticky ends with the plasmid, and insert like the insulin gene. So you need to actually use stuff that is specific from A-level that didn't come up in your AQA biology GCSE. Does it matter how much you write about the importance, like 50-50 in a paragraph? Now, I would say you're generally going to have more AO1, and some people like and the examiner doesn't mind which way you do it. Some people do the AO1. The AO1 is your content knowledge. Some people show off the descriptive statements like, this is the nitrogen cycle. This is what the nitrogen cycle does. Nitrifying bacteria oxidize ammonia or ammonium ions, whatever, into nitrites. Then another species of nitrifying bacteria oxidize nitrites into nitrates. And some people like to hit all of that and then comment afterwards about the importance. But they do want more than one line just saying, yeah, and that was important. So you can either do it line by line, mixed together. You can blend the two together, or you can do them as two separate things. You don't have to separate them. So it depends what your style, what you prefer. So it's important that the first nitrifying bacteria convert the ammonium ions into nitrites. So the second one can convert it into nitrates, and then it's important that that happens. It's important we have denitrifying bacteria, otherwise it wouldn't be a cycle. Nitrogen wouldn't be returned to the atmosphere, and eventually atmospheric nitrogen would go down, and blah, 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 and yeah, all of that kind of stuff. Yes, you could talk about succession. Succession would be a good one with microbes, absolutely. So lots of areas you can squeeze in, and as long as you're pretty creative, you can get them in. So with succession, you've got your pioneer community, Alicia, which is like lichen, and lichen are a fungi. So it depends if it's microbes or specifically bacteria. Lichen are fungi and algae, and kind of like a colony of different little microorganisms, and when they die, they are decomposed by bacteria, and they will form that tiny little humus layer, so their microbes are essential in making bare rock, or really harsh conditions, less hostile, which then allows other pioneer community members like moss and things to put their roots down and stuff like that. So yeah, they make it less harsh. You could talk about succession, decomposition also, and decay. Yeah, all of that kind of stuff as well. Yeah, so we could go with succession. I think we've got some other suggestions. Here's some other suggestions here. Oh, there we go. Tac DNA polymerase. You know, the thermophilic bacteria whose DNA can survive like 95 degrees or 97 degrees of temperature without denaturing. They're really useful in DNA polymerase. So you could talk about that. Insulin production. Eutrophication. What role do bacteria play in eutrophication? I know that sounds like it's in with the nitrogen cycle, but it's a separate subtopic. So do you guys remember what the bacteria do? It's the algae if it's talking about microbes that block the sunlight. But what do the bacteria do to the algal bloom? So the bacteria are going to feed or decompose the dead algae, and they're going to help use up the oxygen inside the water in eutrophication. That's right. They break down. That's right, Betsy. They break down and decompose the algae. So, oh yeah, antibiotic resistance. That's a separate one than immunity, because you get to talk about natural selection there. So you get to talk about your mutations, your variations, and all of that kind of stuff. But remember, we have to keep it A-level. So if you're going to start talking about genes, best to use the word alleles and that kind of stuff, allelic frequencies and stuff like that. So you want to make sure you have a flick through and you look at your antibiotic resistance and you know your specific terms. Oh, also, it hasn't really come up in A-level ever, but it's in your book, is tuberculosis. It comes up in the AS exams. Those students in schools that just do AS, and there aren't many schools that do AS, but some schools offer AS where you do a 1-year course. Tuberculosis seems to come up, and lung disorders in that, and that's caused by a bacteria. Yeah, remove the oxygen from the aquatic species by doing respiration and that. Yeah, I wouldn't bother with all of this. Okay, I wouldn't bother with this one as extra beyond the spec. Just stick to the cholera, really. The cholera covers it with microbes. It's a microbe, so it covers anything on microbes. Covers anything on cell membranes, covers anything on ions. So if you watch the video I did on cholera, there's some other YouTubers who've done videos on cholera as well. Cholera causes the chloride ions to go in the lumen, so anything on the importance of ions it covers. Anything on receptors it covers, because you can talk about how it produces a protein specifically that binds to the receptors, allowing the cholera in and also allowing it to open the chloride ion channels. You can talk about cholera and proteins because it produces that toxic protein. So you want to talk about its primary structure, which gives it a specific 3D shape, allowing it to bind to receptors and that kind of stuff. So it covers so many different ones. It covers osmosis, because with the cholera, you've got the chloride ions leaving the epithelial cells and going into the lumen, therefore increasing the water potential of epithelial cells temporarily and decreasing the water potential in the lumen, causing water to move by osmosis into the lumen, giving the person diarrhea. And then it also covers diffusion if that comes up. And so it covers, because chloride ions then start diffusing from the blood and surrounding cells into the epithelial cells, and then they diffuse again into the lumen, so that you get this pattern of diffusion of ions followed by water down a water potential gradient, like kind of chasing after the ions as they're lowering the water potential. So you've got you cover diffusion and movement across cells and movement within organisms, and any essay titles that have that as well. So cholera is an awesome one. But here's another one for those of you who just want to you can research this. You can just Google this and have a quick read. It takes you 5 minutes and you'll get enough things. Endosymbiosis, evolution of heterotrophic and autotrophic cells. So like basically eukaryotes, evidence that the mitochondria and chloroplasts were once bacteria. Okay, they were once single-celled microbes that came to live in other single-celled organisms, like eukaryotes. And there's evidence. Chloroplasts have 70S ribosomes. That's beyond the spec, but there's a type of ribosome in eukaryotic cells, which is your cells. Animal cells and plant cells have 80S ribosomes. It's the way we describe the ribosomes we have. The ribosomes in bacteria are called 70S ribosomes, and we also find those ribosomes inside chloroplasts and in mitochondria, suggesting they were once bacteria or single-celled organisms like bacteria. They also transcribe their own DNA by binary fission. And they reproduce by binary fission, They transcribe their own DNA, so they don't rely on our nucleus to make their proteins. They make their own proteins by transcribing their own DNA. They have double membranes like a bacteria does. So there's other structures that are single membraned, but mitochondria and chloroplasts, they've got like a double membrane and that kind of stuff. Oh, and they have plasmids or circular DNA. They have circular DNA. So they've got a lot of things like bacteria. So that's called endosymbiosis. You could come with that, but I just stick to cholera. This one came up in 2022. So cycles, importance of cycles. Very unlikely to come up again. If it does, you've got the nitrogen cycle, the phosphate cycle, carbon cycle, but you've also got loads of cycles within organisms as well, inside organisms. So it could specifically ask you a question on cycles within organisms. And there's loads of cyclical processes. Krebs cycle. The light-independent reaction is a cycle with Rubisco, carbon dioxide fixation, and going round and round. So you've got loads of cycles going on there. You've got the cycle of with insulin and glycagon, being released from the pancreas, the alpha cells, beta cells, And so you can spit a load of knowledge there about when glucose level, blood glucose concentration goes too high, insulin's released from what cells, what does it do, what's the process where it converts glucose to glycogen called, what's the opposite process where glycogen's broken down to glucose called, when glucagon gets released. So that's a cycle, controlling our blood sugar level. So you could talk about that as well. Predator-prey cycles outside the body. So there's quite a lot on cycles, but that's come up. So is it going to come up again? Probably not. Water hasn't come up in a while, and water could be worded in a number of different ways. Now, remember the cholera? We can use it for water because it's causing people to die by dehydration. Beyond the spec cholera, it's making them lose water by dehydration. You've got osmosis involved in that process. So you can hit cholera on this. What's the importance of water and regulation in living organisms? Okay, but you can just plan an essay about water basically and why it's important. So why is water important? Muscle contraction as a cycle. I don't know about that one. That one's a little bit more. Yeah, I don't know. I wouldn't go with muscle contraction. It's got to be because, like, your muscles aren't. I suppose muscle contraction of. Yeah, I mean, cardiac cycle with your heart and your myogenic cardiac muscle would be a cycle, but tensing your muscle is not a cycle. A cycle is something that's just going on and on and on. Your blood sugar's going up and down, up and down, up and down throughout the day. Every hour or every half an hour, every 10 minutes, your blood sugar is fluctuating like that. It's being controlled by a negative feedback mechanism. But muscle contraction is voluntary, but there are involuntary ones that are cyclical, like your heartbeat. So yeah, you can talk about the cardiac cycle there. But importance of water. So why is water important? What would be your 4 topics to do with water? There's like 20 topics you could dip into here. The great thing is if you revise the photosynthesis, photolysis, but we can talk about respiration or photosynthesis and the role of water in it. So we know in photosynthesis, we can then chat about hydrogen ions. We can chat about supplying the electrons for the electron transport chain. That comes in photosynthesis from the water, and photolysis supplies the hydrogen ions for chemiosmosis. Then you can chat about chemiosmosis, why that's important. If we didn't have water, we wouldn't get chemiosmosis. The plant wouldn't make ATP and NADP reduced because it wouldn't have hydrogen ions to reduce the NADP and all of that. So you keep referring when you want to say something's important, say what would happen if it wasn't there. So like if there was no photolysis in photosynthesis, what wouldn't happen? Well, there'd be no electron transport chain, so there couldn't be chemiosmosis. So there couldn't be ATP, there couldn't be NADP reduced. They couldn't go on to the light-independent reaction, and plants couldn't make carbohydrates. Death of plants. So you need to put all your detail and then link it to the question. You could do respiration. What's the role of water in respiration? Yeah, oxidative phosphorylation, Betty, that's right. And osmosis. Yeah, properties of water. That's right, Lucy. So you can come with your properties of water. It's good at dissolving metabolites. It's a solvent. So it can dissolve metabolites like amino acids and glucose. And if we didn't have water to dissolve amino acids and glucose, then we couldn't transport any of these products that are needed for respiration and stuff around our body. Yeah, oxidative phosphorylation. Remember, oxygen, what's water used for in oxidative phosphorylation? The oxygen combines with the hydrogen to form water, but I don't know whether that's probably a bit of a stretch, that one, but yeah, it's there. But whether or not they would like that one, you need the formation of water, I suppose, because you need the oxygen to join with the hydrogen and accept the electrons. Otherwise, oxidative phosphorylation doesn't happen and you don't get ATP without the formation of water. Water is also needed for all of your biological molecules. Why is it needed in biological molecules? So you could do a whole paragraph on biological molecules. Why do we need water for your dinner? You guys have probably just had dinner. Why do you need water? Hydrolysis. Yeah, that absolutely. So if you don't have, and this is where the importance bit comes in, if you don't have water, you can't hydrolyze polymers like carbohydrates or proteins. You don't get access to the amino acids, you don't get the glucose. State what it is you don't get, but in that you get to show off your knowledge and say, oh, you can't hydrolyze the glycosidic bond, so now you're scoring marks all the time. Yeah, so you get to talk about digestion and hydrolysis, but you've got to keep it A level, so you're scoring the marks. Every time you say something like glycosidic bond, peptide bond, that's scoring you points, right? You think of it like each paragraph as like a 5 or 6 marker, so you need all those like nice keywords. But don't go too much off. If you're doing digestion, don't start going into co-transport across the ileum with sodium ions, because that's not really related to water. So don't go into all of that. Don't try and show off too much. And start getting irrelevant. Now, if you write 1 or 2 irrelevant lines on each paragraph, they're going to allow it. They've said the exam board, they'll allow it. It's when you start saying like whole paragraphs that are irrelevant, they're like, oh, you're going to lose a couple of marks. They're going to say they've got quite a bit of irrelevant material. So, what other essays? Hope everyone's revision is going well. No, don't do the essay first. Most of the YouTubers, again, I don't know. I've kind of given it some thought, but I wouldn't do the essays first. The reason... yeah, don't do the essay first. Don't do the essay first. Some people, some YouTubers have said, like that I've seen comment on it, have said read the essay first, then go to the start and work through the exam. And what they're saying there is, if you know the essay is on ions, and as you're working through the exam, you might get something called hemoglobin, Fe2+. You'll be like, oh yeah, I can talk about hemoglobin in my essay. So they say read the essay first, then go to the start of the exam and work through, and subconsciously, as you're going through, your brain will be like, oh yeah, that's about protein. I can include that in my essay on proteins at the end. And you'll get a picture, maybe. But some people, it might just distract them reading the essay first. So it's your choice, really. I guess, you know, it's your choice. But yeah, you could do the essay first if you want. But they give you 40, they recommend 45 minutes for the essay. But yeah. Do you do it first? Who knows? Right. So some of the other things people mentioned about water was Lucy said osmosis. So obviously big chunk to talk about osmosis. We talked about hydrolysis, mass transport. So you could work in mass transport from the source to the sink. If you get a question on osmosis, now there was one question in the past about 12 years ago on osmosis, the importance of osmosis in organisms. And of course, osmosis, we get active loading at the top of the phloem in the sieve tube. We actively load sucrose and sugars in there, and that lowers the water potential, and then we get the osmosis of water. So you could talk about water there. Water is important for mass transport. It's needed. You could then go into that detail, or you could talk about mass transport in plants. The transpiration stream, cohesion, tension. Again, we need water for that. We need the evaporation of water to create negative pressure. You could get an essay on mass transport as well, or on transport systems. So you've got the heart. You've got mass transport is lungs, breathing it, ventilating your lungs. So you'd need to know intercostal muscles, which ones, is it the external, internal, that kind of thing. So you'd need to know how we ventilate the lungs. For mass transport, your 4 would be lungs. You've got the blood. Circulation of the blood is mass transport. How do we create a high hydrostatic pressure? That type of stuff. You've got formation of tissue fluid, obviously, as well in mass transport. And then the two plant ones as well, which are cohesion, tension with water up the xylem, and movement of sugars down the phloem, which involves mass transport by creating a high water pressure. Yeah, I don't know about doing the essay first. I wouldn't do the essay first. Just check in. Let's have a look. Yeah, here are some. These are ones to talk about with water. Again, I would just do cholera for water. So the cholera one can really pull it out for most of these because it causes the loss of water. And that's important because we need water to stay hydrated and for all those things. We need it as a solvent. We need it to dissolve metabolites and all of that. Just in case anyone's interested. But I again, as I say, I would just learn like 2 or 3 of those extra spec things. I mean, with the extra spec things, you kind of like, you can get 20 marks without that. So you can get up to like, which is a huge percentage of your final grade. They say the essay is 10% of your biology. So, yeah. Can life exist without water? So this is just some extra spec stuff. Just saying that we know all those funky things about water, like it's a great solvent, it's a great metabolite. Well, apparently on planets like Venus, where it's very hot, at certain temperatures called the critical point, or they can become certain gases like carbon dioxide can become what we call a supercritical fluid. There's an extra spec thing. And it means it's a gas that now has the properties of a liquid as well. And so it can act as a solvent. And apparently, if carbon dioxide becomes a supercritical fluid and it needs a special temperature, 305 Kelvin and 73 atmospheres for it to become this supercritical fluid, then apparently enzymes are more stable and their active sites even more specific. So less likely to be inhibited and that kind of thing. So less likely to undergo unnecessary side reactions or to suffer from competitive inhibitors because their active sites become specific. I mean, they could still be affected by non-competitive at the allosteric site. But yeah, that's an extra spec bit. But to be honest, once I know that water links to cholera, I'm just going to use that. But for those of you that don't want to get funky, yeah, that's another one there. Oh yeah, there's an extra spec bit. Now, a variation came up fairly recently, I think, but variation's a disgusting one if it comes up. It's one of those ones where you're like, oh my God, what do I talk about? So these are the themes for variation, and you can literally talk about any 4 or 5 of these. So how does protein create variation, or how do variations stem from proteins? So you think of primary structures. Primary structures inform your tertiary structure, create like variation in active sites. So then we can start talking about enzymes, active sites being specific and why that's important. Variation is created by mutations. So you can talk about mutations and why they're important. If you get a specific mutation and it changes the nucleotide base sequence, it will change the primary structure, which will then change the tertiary structure of the protein and give it a different shape. And potentially that could be advantageous and blah, blah, blah. We could talk about natural selection, meiosis, how we actually get variation from meiosis. Why is it important that we have variation? Why is meiosis important? Well, animals that sexually reproduce show variation, and why is that important, basically? The more variation, genetic variation you have, or more variation within a gene pool, the higher the chance of surviving a change in the environment, which is relevant to climate change and all of that kind of stuff. Biodiversity, how do we measure variation? So you could talk about biodiversity. So variation was also a 5 marker. So yeah, I don't think variation's coming up. That's a good point, Alicia. So, but you know what? Just in case, I mean, you're going to get 2 essays. So the chances are one of the good ones is coming for you and you're going to get a nice one to talk about. But just in case, imagine you've got variation and then something else that you hate. You'd be like, ah. So yeah, what I would do, I wouldn't write an essay on variation for that reason, because it's come up in the last few years and it's already come up in paper 1. I'd just say to myself, Worst comes to the worst, if I was forced at gunpoint to write an essay, can I come up with 4 topics to talk about? So biodiversity, you've got the index of diversity. You need biodiversity is decided by variation in the species and the abundance, the number of different species and the abundance of each species. It takes both into account, and you get a number. More stable ecosystems have higher variation, more likely to survive changes and stuff like that. But yeah, it's all there kind of thing. So cool. Yeah, paper 1, a lot of people said paper 1 was hard, but people prefer paper 2. So it's all really good practice for you. All of this is great practice for you for when you go to uni, like finding some exams hard and some easy. It's all great. It's all part of the learning game. Like you're in it. You're in there. Like it's great. And you guys missed out on your GCSEs, so this is fantastic for you in the long run. All of this is really good for you in the long run because you missed out on having to do GCSEs. So when you go to uni, the exams come really thick and fast, but they're not as hard as the ones you're doing now. So even if you don't get the grades that you want, you'll be in the same boat as lots of other people will be missing their grades and their perfect uni place, but you will be going to university. With the grades you get, you'll be able to go to university and have that experience and study, and the exams at university are more like easier in some ways. More detailed, but kind of easier. Yeah, I heard it was all paper 1 immunology. Yeah, so like take that for instance. When you go to university, you'll do like 3 months of study, 4 months of study, and then you'll have a set of exams. You might have 8 exams in exam week. And with one lecturer, the whole course, 5 hours a week, you'll have been doing immunology for that semester or term or whatever they call it, that section. So you'll just have an exam on immunology. And then, so it's like end of topic tests kind of thing. Yeah, that's kind of like what it is. But they're a bit fat. There'll be an essay on, there'll literally be an essay when you're at university on many of the topics. So AQA is great because it has this horrible essay and it prepares you for uni, because every degree you do at uni, you're going to be writing essays. So the exam boards have said they intend to lower the grades this year. They said they've been really generous because of COVID and have been like allowing people, and they said they're going to lower them. But the exams weren't easier the last couple of years. They didn't seem easier, but people are saying, oh, it's going to be more like 2019. But I don't think they were particularly easy. So the point is, they could never lower the percentage massively or make it much harder. So they can't really change it a lot. It just doesn't look good. So I don't think they'll be massively lower this year. Although they did say they were making the exams harder. So maybe you guys have already had the harder exams. Then they will lower the grade boundaries. They need to have it. They already know before the first kid has written one word on a paper, they know the rough percentage of A's, stars they want and A's. So they just adjust the grade boundaries accordingly. Yeah, physics is always hard. The great thing, Alicia, with physics is that a grade in physics is worth, like even a low grade in physics is worth more. Like, for instance, in teaching profession, like at my wife's school, they got one physics teacher out of like 20 teachers in the science department. They've only got one physics teacher. So it's like if you walk in and you're not crap, then you're going to go really far. You can literally hold the school hostage and be like, yeah, I'm going to leave unless like you make me head of year in 2 years or something, or you pay me more. So physics is one of those subjects. It is super hard. So yeah, your grades can be worth more. Variation, you're not getting variation, but if you were, that's a little bit of extra curricular knowledge. You can rewind this if you really want to take the chance and memorize that. But basically, most species, they can measure how much diversity there is within the species. And apparently the most diverse species in the world is the split gill mushroom, which is a fungus, binomial name, Schizophyllum commune. So that's its binomial name, Schizophyllum commune. So this would be beyond the spec if you've got something on variation. And it has a nucleotide diversity of 20%. You could just call it the split gill mushroom and screw the binomial name. But the binomial name is A-level biology, isn't it? So, and what does that mean, 20%? It means if you take any two mushrooms, 20% of their nucleotides are different from each other, which is a huge amount of variation when you think that we say stuff like, oh, we're 98% similar to a chimpanzee. Well, this mushroom isn't even 80% similar to another member of its species, so it has the most diversity. Therefore, it's been found absolutely everywhere in the world except for like the Antarctic. So it's like can survive any habitat because of its great variety and its genetics. Nucleotide, that's diversity, I think. Yeah. Yeah, so importance of variation. Yes, that's a good point. So we could say the species is hyperdiverse, and then we would link that to say this enables this species to survive like changes in the environment, like climate change and things like that. So while other organisms can't survive, there's more likely that this one has a protein, one of its members will have an allele that will allow it to survive many different changes and diseases. So the smaller your diversity, take cheetahs. Cheetahs aren't diverse at all. They're basically all seriously related. All their genes are the same. So if any disease comes out that kills cheetahs, it's going to wipe out and make them extinct, basically. So they're less likely to be extinct. So having that variety in your genes means you're less likely to become extinct, more likely to survive changes in the environment. So that's why I would link that to the importance, just to point that out. Yeah, you could link that to DNA as well, you see. So you could link that one to DNA. Might do. Oh, yeah. So microscopes, just in case. I don't think they're going to ask about microscopes. Do you know what? They've asked about microscopes about 16 years ago. I don't think they're ever going to ask about microscopes again. But these are the topics I would focus on. Different types of microscopes. So do you know your light microscope, your electron transmission electron microscope, and your scanning electron microscope? You wouldn't have to talk about them. You can just say microscopes have allowed us to understand about cell organelles, and then you can describe cell organelles. We wouldn't even know cell organelles exist without microscopes. So now you can talk about the function and structure of cell organelles as a whole one of your 4 paragraphs. They've also allowed us to understand what a tissue is, a group of cells working together, and then you could talk about a certain tissue in the body. They've also allowed us to observe mitosis and understand how cell division works in the different phases. We can see the different phases of cell division, PMAP, prophase, metaphase, anaphase, telophase, using a light microscope. You can actually see them. So they've allowed us to understand that. So I would then, you just describe the phases and what happens and keep saying, yeah, we can only know this because of the microscopes. Things like that. So even Carl Woosey's finding out that RK bacteria are different to eubacteria in the domains, noticing differences in structures and stuff. So if you get a microscopes question, you can talk about literally tons and tons of stuff that we can observe with microscopes. Anything you can see under a microscope, you can talk about and say it's important, otherwise we'd have never known this. Carbon dioxide's been asked about twice in the past. So What, the microscopes? Do you think the microscopes one is pretty disgusting? Is that the microscopes? Let me tell you, microscopes is probably okay. Let me just expand on that. I don't think it's coming up, but it's actually, if it did, it's actually probably one of the easiest, because you don't that doesn't mean you have to remember all your microscopes jargon. You might be like, oh my god, I don't know nothing about microscopes. The question's saying why they're important. It's not saying tell me about how microscopes work. It's saying discuss the importance, unless in the question it says discuss how microscopes work and the importance of them. So you could talk about a separate topics to do. Sorry, my WhatsApp. Someone's WhatsApping me. You could talk about a separate topics. So when I say talk about tissues, you could talk about the structure of the kidney, you know, the loop of Henle and all of that. So we can look at that under a microscope. So you can describe the structure of the kidney and talk about its function, and say we would have never learned the structure of the kidney without microscopes. So you could do a whole bit on that. Muscle structure and the striations, you know, the H zone, the I zone, the A band, whatever, the I band. If you know that really well, if that's something that you're like, yeah, I could do 5 marks on that, talk about the structure of muscles, because we wouldn't know about that without a microscope. But if you know it really well, don't talk about it if you think, oh, I'm a bit crap on that. Cell structures, you all know really well. So you definitely are going to get a whole paragraph on talking about the endoplasmic reticulum, mitochondria, ribosomes. Obviously, ribosomes you can't see unless you've got an electron microscope, but you can talk about all of those, basically. And then mitosis and the mitotic index, and then you can relate that to cancer, the importance of basically, if someone has a high mitotic index, it might suggest that those cells are dividing too rapidly, and that could link to cancer and things like that. So also, we can observe using microscopes bacteria and fungi and other single-celled organisms and learn the difference between them and say, hey, these ones have chloroplasts, or these ones have a cell wall. So it can also teach us about variation between species and taxonomic differences. So, like, for instance, we can observe with a microscope that a fungi, a plant, and animal have nuclei, so they're eukaryotic. We can explain that, and then we can do a bit of classification and say, however, bacteria don't have a nucleus. So anything you can think of that you could look at through a microscope, basically. Fertilization, sperm and egg to form an embryo, but that would be more GCSE, so I'd probably steer away from that. Restriction endonuclease and ligase and PCR. I wouldn't talk about that for microscopes, because I probably wouldn't talk about that for microscopes, because you probably can't see the enzymes. They're probably too small, like viruses, too small to even see through a microscope. Protein synthesis. Could you talk about protein synthesis as one of the processes you could observe? Mitosis you can observe, but protein synthesis is happening on a ribosome. We can just about see ribosomes with an electron microscope. You probably couldn't observe protein synthesis with an electron microscope. But you could discuss different microscopes if you know your different microscopes, like your resolutions and things like that. You could discuss one of those for a paragraph, then the cell structures for a second paragraph. Then you could discuss the kidney or the structure of a leaf. If you know your structure of a leaf, your palisade cells and all of that stuff. Then you could discuss mitosis, cell division. That's a separate process. And you could do classification, or you could do cancer, identifying cancer and things like that. But yeah, that could be an essay that you might want to steer clear of. Oh yeah, carbon dioxide. Carbon dioxide and its effect on organisms. So can you think of 4 places in your book where carbon dioxide turns up? Can you think of 4 processes or topics where CO2 is mentioned? And literally then wherever it's mentioned, it's important. So the light independent reaction, it reacts with RUBP. That's right to form. Is it 2GP it forms first and then it, yeah. So if we didn't have carbon dioxide, so when we're doing important, brilliant photosynthesis, thank you, Pretty and Alicia, yeah, gas exchange. Yeah, photosynthesis is a limiting factor, great, so that's going to affect crop growth and that. But remember, when you're talking about limiting factors, it can be difficult to make that A level. Whereas you definitely know if you hit the light independent reaction, the examiner can't say, oh, this person's waffling some GCSE stuff. I know limiting factors turn up in A level, but they don't want GCSE knowledge, so try and hit the stuff that's more unique. Yeah, hemoglobin, yeah, and the Bohr shift. Yeah, that's right, Lucy, we've got that Bohr shift. When you're respiring and the carbon dioxide causes the sigma curve to shift to the right, doesn't it? So the hemoglobin changes shape and has a lower affinity for oxygen at higher partial pressures. You could do the whole paragraph on that, definitely. I definitely do the light independent reaction because you can always stress the importance. Like, if carbon dioxide didn't change the shape of hemoglobin, then our hemoglobin wouldn't adjust and pick up oxygen for the respiring muscles, and we wouldn't be as efficient at respiration, and we wouldn't be able to be as active as we are. We wouldn't be able to exercise as hard or run as hard, and that would affect our survivability. So that's an important thing. In the light independent reaction, if carbon dioxide doesn't fix with ribulose biphosphate, RUBP, in carbon fixation, then we don't form GP, and plants can't lock the energy from the sun. They can't take that energy. Remember, the energy from the sun is held in the ATP that they made in the light dependent reaction. They can't take that energy from the sun without the GP and turn it into carbohydrates and proteins and things. I would check the syllabus for that. I mean, I've always taught GP and that, but recently people have just been saying, right, glycerate 3 phosphate once. Then, yeah, that's the best thing, Alicia, to do. I've never massively taught that, but recently a lot of YouTubers have been saying, oh, if it's not in the spec, you can't use abbreviations. Like, I've always marked papers and given people marks for abbreviations, and so are probably most of your teachers. So that's something that's really being hyped up at the moment. So yeah, that's what a lot of teachers, everyone's hyping this up at the moment, saying you can't abbreviate. But like, if you read through the spec, and in the spec it refers to it as GP, in the specification, then you can call it GP. Yeah. As long as it's in the specification and it says GP in capital G, capital P, you can call it GP. But if it says glycerate 3 phosphate, then damn. So, yeah. You could flick through the spec. Do you have a spare half an hour before Wednesday to flick through your spec just on screen, go down, or and just be like, yes, write down all the ones that appear in the specification with the abbreviation, because then you can use them. Yeah. So you've got chemoreceptors. You could do a whole chapter on that if you know your chemoreceptors, and then your, which nerve? Is it parasympathetic or sympathetic nerve? So it's chemoreceptors. Do you know the location of the chemoreceptors? They detect changes in the carbon dioxide level in your blood. And do you know their location? You're going to score marks for saying like aorta, carotid artery. And they detect the carbon dioxide. They send an impulse. Is it to the medulla? And the medulla sends an impulse down the parasympathetic or sympathetic nerve. And they release a certain neurotransmitter that acts on your heart and increases your heart rate, meaning that you can get rid of the you can ventilate the carbon dioxide. So like there's a whole chapter there's a whole section on that. Light independent reaction we just talked about, definitely. That depends if it's the importance of CO2 in or just generally. You've got crop yields. Distribution of organisms. Well, we're talking about global warming and how that's going to affect the distribution of organisms and plants like carbon dioxide because it's a limiting factor of photosynthesis, that kind of thing. You couldn't get a vague link that carbon dioxide has caused global warming, okay, and that has increased the temperature of the Earth, and that will affect enzymes. The higher the temperature of the Earth gets, lots more enzymes in organisms are starting to denature, and those organisms could become extinct. So remember, human enzymes are kind of okay, right? They operate at 37 degrees, but that's not all organisms' enzymes. And then you can talk about ventilation and stuff like that, how we get rid of carbon dioxide, how we breathe out. I mean, this will be a good one, right? This has come up twice, actually. Twice in the last 15 years, polymers. That's not how you spell structure. What do you guys think for polymers, and how is their structure related to the function? What would you make your 4 paragraphs on? What 4 polymers would you pick? That's good, Lucy. Good question. Lucy said, how do you make ventilation? I can't even speak properly. How do you make ventilation A-level? So at GCSE, it's the terminology. So what do we call this region at A-level? And which muscles contract at A-level is different than GCSE. So at GCSE, we just say I can't even speak. At GCSE, we say intercostal muscles contract and the diaphragm contracts and that. But you have to know whether it's the external or internal intercostal muscles that contract during ventilation. That's a good point. Yeah. So Alicia, I think you covered that. That's let me come back to that, Lucy, as well, about the carbon dioxide. Alicia said starch. So yeah, you definitely can do starch. Now, I can't remember in AQA if you guys do amylose and amylopectin, then you can do a whole paragraph on starch, amylose, amylopectin, why it's a good storage molecule, why one is more heavily branched, or one is branched, one is not branched. That kind of thing, why they're insoluble. So how does their structure give them a function or whatever? You've got cellulose, which you could do a whole other paragraph on. You've got glycogen. Yeah, this one's dead easy if we get this. Glycogen, again, you need to get all your alpha beta glucose, your stuff right, branching, talk about why it's a good storage molecule again. Remember, DNA is a polymer. Remember, RNA is a polymer as well. So you only need 4 of these, but I'd get a bit of diversity there rather than just doing the kind of like glycogen, cellulose, and starch, which are all glucose-based with glycosidic bonds. I want to get my phosphodiester bonds. I'm trying to score, if all 3 paragraphs are talking about glycosidic bonds, I'm not getting as much marks if I can maybe talk about phosphodiester bonds in DNA and RNA, and of course the differences between DNA and RNA. Yeah, you do amylose and amylopectin for AQA, so you can make that starch one nice and fat. Talk about how it's related to its function. Cellulose forms microfibrils, the cross-links from the beta-glucose. Beta-glucose, every other molecule is reversed 180 degrees. It's insoluble, blah, blah, blah. It's a structural one. All of that sort of stuff, yeah. The fact that glycogen and sort of amylose and amylopectin are insoluble means they don't have an osmotic effect and cause cells to swell up. So you can talk about all of that stuff. The DNA, there's a lot to talk about with DNA and how it's structured, the double helix, the sugar phosphate backbone and stuff, how that's related to its function. You can talk about the complementary base pairing of the monomers, the bases, and that kind of thing. You've got to be careful with lipids. This is a good point you brought up, Alicia. Lipids aren't actually polymers. Remember, a lipid is not a repeating unit like an amino acid, amino acid, amino acid. A lipid is one glycerol and 3 fatty acids. So it's not a very big molecule in that sense. Yeah. Fatty acids are quite big molecules, but the overall it's not a big chain. So skip the old triglycerides and phospholipids in that one. No, that's cool. That's cool. Yeah, you've got all the bonding as well. If you do protein, I haven't even written protein on there. It's like the best one, isn't it? The best polymer to talk about because there's just so much, all the structures and everything, and how that relates to its function. That's the best paragraph to talk about. But you need to cover those 4 relevant paragraphs if you want to get up to 20 marks. Remember, you've got to get 4 relevant paragraphs and A-level content to get 20 marks. Now, Lucy said, what's the extra content for the carbon dioxide question? Good point. Can we work cholera into carbon dioxide? I don't think we can work cholera into carbon dioxide. But I did have that, I did have a weird... This one, you could work into carbon dioxide. And you could say, hey, carbon dioxide could be even more important than we first thought. Carbon dioxide may replace some of the functions of water on other planets. For example, when carbon dioxide reaches its critical point at 305 Kelvin and 73 atmospheres, it becomes a supercritical fluid. I can't spell super, that says supra. It's supposed to say super. Oh, jeez Louise. But there's probably other better ones. I will have a look at some other ones. I need to find another one that will cover like loads of other topics as well. Yeah. And when carbon dioxide enters a supercritical phase, apparently it's a more stable fluid than water for enzymes to exist in. Their active site is more stable in a fluid of carbon dioxide, apparently, and their binding site is more specific, and so less chance of inhibition and unnecessary side reactions. Just for a little extra bit there. Yeah, we did that. Yeah, so that one's lovely. There. That's a kind of disgusting question. Relationships and interactions between organisms. So, pretty, year 2. And remember, you don't have to do year 2. Any 4 subtopics. Doesn't matter. But if you wanted to talk about year 2 and polymers and their importance, you could get into the DNA technology like PCR and stuff like that and link that. But you just got to keep linking it to the fact that it's a polymer. So, and RNA and stuff like that. You've got to link it to the fact that it's a polymer. But yeah, you can bring that stuff in from your electrophoresis and all of that. But you've got to cut. It's more difficult to link that in the year 2 polymer stuff. Yeah, speciation. Nice. Yeah, great. So when they, and of course, whole paragraph on speciation would be awesome. So you've got your reproductive isolation, geographic isolation. You've got your natural selection occurring differently, different environments. By the way, at A-level, in your essays and stuff, talk about alleles rather than just saying genes all the time. Talk about alleles and stuff like that and frequencies. And yeah, and then you've got to talk about they can no longer interbreed and all of that. So yeah, speciation, you could get one there. Courtship behavior, brilliant. Yeah. If you can talk about that for a whole paragraph, courtship behavior, you might find yourself writing like 2 lines and going, what do I talk about here? Like, is there enough meat to get... Got to think of each paragraph as a 5 marker, like it'd be the end, like have you hit 5 kind of key words and key points. It's kind of to think of it. Now I know you're going to link it back to the question as well with your AO2. You're going to apply it to the actual question, what's the importance of courtship behavior, which is probably where you can get the marks, because actually if you talk about courtship behavior and what's the importance, really important there, because otherwise you get you don't get fertile offspring because they don't recognize they're the same species and all of that kind of stuff, and all of those kind of things. So yeah, there's loads more on courtship behavior that you can link back. Symbiotic relationships, absolutely, but can you think of any with enough meat to talk about on symbiotic relationships? So yeah, you could do that one. That mycorrhizae. What was that movie about the fungus that killed everyone and the little girl and the bloke walking around? That had that fungus in it. So yeah, that was the fungus in that they had killing everybody. But yeah, you can talk about that. That's it. The Last of Us. What other things involve organisms? Thinking ecology, you've got, nice one is succession. You guys can all get the 5 marks on succession. Great chance to get that in. How's that? Interactions between organisms. You've got your pioneer community, then you've got your climax community. There's 2 marks right there, explaining how they change the conditions, how the biodiversity increases, the biomass increases, and then you get like your climax community with one species dominating and all of that. And the biodiversity decreases a bit and it becomes stable. So yeah. Inheritance. If you wanted to dare go to inheritance and discuss how we inherit dominant and recessive alleles, dihybrid inheritance, if you really wanted to get disgusting about how that's a relationship between the parents passing to the offspring. But this is a great time for cycles again, isn't it? Nitrogen cycle, all the organisms and interactions between them in the nitrogen cycle. And you've got your symbiotic relationships, which Bridget mentioned. What is the name of the bacteria that live in the root nodules of leguminous plants? So do you remember your bacteria? So that's one you could probably talk about quite a bit, because you can use the word nitrogen fixation, nitrates, talk about them becoming amino acids. Name the bacteria that live in the root nodules that do the nitrogen fixation and the symbiotic relationship. Nitrogen fixing bacteria, are they rhizobium? Or I think you actually have to know the name. So for A level, you'd want to bust out the actual specific name. Otherwise, they might just say, hey, GCSE triple kids, no nitrogen fixing bacteria. So there's actually quite a lot on this one, because you could do loads and loads and loads. You've got antibiotic resistance. You've got gene technology, haven't you? Because you could go into your whole... I don't know if you guys are really cool with gene tech. If you're good at the gene tech, the in vivo stuff and plasmids and the restriction endonucleases and all of that, and the ligase. If you're good with all of that, that's an easy 5 marks there, because we're using our relationship with bacteria. We're using them to produce proteins for us. Do we need to know? No, you need to know the names of all the bacteria in the nitrogen cycle. As far as I'm concerned, I think you do need to know the names. Now, would they dare to give you an essay on the immune system? I hope not after paper 1. I think people would cry if they got that on an essay on the immune system. There's a nitrogen cycle. I'm sure for AQA you've got to know all the names for the nitrogen cycle. So 306, whipping out the book. Let me just double check that. I don't want to be telling you to go and learn the names. Maybe I'm talking out my bottom and you don't need to know the names. Actually, that would be extra because I swear you had to know the names. I'm looking in the book and I need to check my spec. It's a bit embarrassing. I didn't mention any of the names. Oh my good gosh. Actually, yeah, it doesn't appear using the old AQA book. It doesn't appear like you need to know the names. So, yeah, it doesn't appear like you need to know the names. I swear, maybe I'm just like, that's my OCR brain switching in. They need to know all the names in OCR. You definitely need to know the names. You need to know the names in somebody's A level for the nitrogen cycle. Wow, you don't need to know the names. Okay. If you don't need to know the names, then that's beyond the spec. That would be beyond the spec information. Now, they mention them in the CGP. They give you all the names in the CGP. So if you have AQA CGP, they give you all the names. So you've got rhizobium bacteria, but I can't guarantee that's beyond the spec. I need to go look at the spec and check that. You've got rhizobium bacteria that turn nitrogen straight into ammonia, then nitrates, and they live in the root nodules of plants. They're leguminous plants like peas and beans, and that's why they're high. That's why baked beans are really high in protein and stuff, because they have those rhizobium bacteria living in their roots. And then they have you've got nitrifying bacteria are known as nitrothomonas. They turn the ammonium ions into nitrites, and then you have nitrobacter that turn the nitrites into nitrates, and they're both nitrifying bacteria. So, you don't need to know the names. That's beyond the spec. Let's write that down. If you get anything on the nitrogen cycle, nitrogen-containing compounds, or let's write that down. They are beyond the spec. I just checked. So, you're right. So, myco, so that, yeah. No individual names. So, yeah, you're right, Lucy. So they would be beyond the spec, but would it be enough? I reckon it would be enough. But in the nitrogen cycle, you can look in your CGP. If you own the CGP, they're all there. You've got rhizobium, are nitrogen fixing in the roots of leguminous plants. So they fix nitrogen into ammonia. So you do need to know what nitrogen fixing is. So you need to know that they fix them. That's a bacteria right there. This can come up also in microbes. You could mention this as beyond the spec. It's because you don't need to know the names. So this would be beyond the spec. You then got nitrothomonas convert sugar. They don't convert sugar. They convert ammonium ions into nitrites, NO2, for those of you that do NO2, whatever the hell it is, NO2 minus or whatever, if you're doing chemistry. And then you've got nitrobacter convert, well, oxidize is probably a better word than convert. Oxidize nitrites to nitrates. So would that be enough? Maybe you're using 3 beyond the spec names there. Yeah. It's not even on the spec, so it just says it's not on the spec. So, and then there's denitrification. There's bacteria that do denitrification. I don't think they mention their names. Denitrifying bacteria, but yeah. You've got the phosphate cycle and other cycles. But yeah, it's a pain. So my recommendation is, before you go in your exam, your essay revision you got tomorrow, for your essay revision, you just want to go in going, right, so the bit to revise, to memorize, is just the 4 or 5 chapters for each essay title. And that way you've got 4 or 5 and you're like, yeah, if it comes in, I'm getting 15 to 25 marks. I'm there. Like, rather than writing out the perfect essay. Of course, it's wonderful if you've written out a 25-mark essay from someone that you've got on energy transfer. But if it doesn't come up and you kind of don't know the others, then you're sort of shafted. So it's better to kind of do a broad tactic. Again, this was another one, why nitrogen-containing compounds important in biology. Essentially, that's protein, and DNA contains nitrogen. So you can talk about why DNA is important, whole paragraph, why proteins are, why amino acids are important, why anything that's protein, enzymes contain nitrogen, hormones contain nitrogen, the nitrogen cycle, nitrates, active transport route, all of that. So loads to talk about there if that comes up. You could talk about anything that has nitrogen in it. So you're looking at the nitrogen cycle, you're looking at different types of proteins because they contain nitrogen. So you can talk about them as different topics if you're pushed. And then you've got DNA, and you can talk about RNA as a separate paragraph to DNA and its importance. Can't think of anything else. There are like even more essay titles. So has anyone got any questions on anything else before I chip up? I might do a general just live tomorrow on like last minute, just not essay based. I might do one in the daytime actually. There are like, I've got the 40 essays for the last 20 years, so that they've asked. And they only come up like with the same one. They've only come up the same, like a couple of them come up again and again, but most of them, they come up with different ones. But you can generally repeat the same kind of stuff because it's all part of the same thing. So, anybody got any requests for any topics? I'm gonna go live tomorrow during the daytime. So if anybody's got any requests for any topics, yeah, it's just about like really just, they're not detailed essays we've gone through, but just about looking at the topics and going, right, if I know 4 of each, I can be confident that something's coming up, and just how to think to cover 4 different topics. You literally can work anything into it. So, yeah. Yeah, so no, that's not random. Hemoglobin and the importance of ions. How ions are important in hemoglobin. So, yeah, well, there's quite a bit on ions in hemoglobin because you've got the Fe2 plus ion. There's quite a bit about hemoglobin, actually. Let me check the AQA spec, how much detail they go into, because that's a good one. Again, I just stick with the cholera and the chloride ions as my bonus. But let me see how much detail, because there's quite a lot with hemoglobin. But I need to check, because AQA is actually slightly less detailed. That doesn't make it easier. Slightly less detailed than the 2 most popular A levels for biology. The 2 exam boards are OCR and AQA. Most schools don't do Edexcel at A level. So there isn't a lot. It's in the protein chapter, isn't it? So you just need to know, right, so what you can do, if you're talking about ions and their importance, you can just say that the ion, Fe2 plus, is found in hemoglobin, and then you can talk about hemoglobin's whole role and why that's important. Let me just see if you have to know whether that's beyond the spec, all the details about Fe2+, because it might be in your biological molecules with inorganic ions. So it's in the biological molecules. They don't mention a lot at all. So in AQA, you just need to know that hemoglobin is a large protein, carries oxygen around the body. It's made up of 4 polypeptide chains, each with an Fe2+ in the center. And it's the Fe2+, the ion, that attracts the oxygen. So essentially, you've got your polypeptide chain with your Fe2+. There's your ion, Fe2+. And then you have your polypeptide chain wrapped around it. That's the worst wrapping around of a polypeptide chain. And the ion isn't that big compared to the polypeptide chain. Just remember, like, ion is like one atom with a couple of electrons missing. So it's like, there it's the size of an entire 10,000 atoms. So yeah, that's a bit big. But essentially, remember that oxygen will bind with the ion 2+ ions. So they're going to bind to form like iron oxide, giving you your blood that red rusty color, which is why your blood looks red. But essentially, all you need to know is that Fe2+ is what binds with the oxygen to form oxyhemoglobin. Yeah, cool, Betty. Yeah, no problems. Yeah, I'm going to jump in on tomorrow. I'm just trying to think what time tomorrow. Well, sometime in the daytime, but you can always click on and rewatch and see what I've gone through. But if there's any topics people want me to talk about. Yeah, so I would say it's literally in hemoglobin. But for the paragraph, Lucy, for the whole paragraph on that, you won't get like lots of marks just for that. You'll need to explain what hemoglobin does. Right. So what do we need to know? So here's what you need to know. Fe2+ reacts with oxygen and temporarily forms Fe3+. Being a transition metal, it can change oxidation states and forms Fe3+, while bound to the oxygen. And then what happens is it becomes Fe2+ again when the oxygen, whoopsie, is released. So I guess the fact that it can be Fe2+ and Fe3+ makes it a really special ion. It's a transition metal, so it can have variable oxidation states. But you don't need to know that, but you just need to know temporarily it becomes Fe3+ once it's bound to the oxygen. And then it releases the oxygen again, and it goes back to being Fe2+. And you just need to know that it's at the center of a polypeptide, and hemoglobin is made of 4 polypeptides, so it has 4 Fe2+ ions. Hence, one hemoglobin molecule can pick up 4 oxygen molecules. So 4 heme groups, or 4, there's sugar. 4 polypeptides in hemoglobin, so you'd want to say that. There's 4 polypeptides in hemoglobin. Each contains Fe2+, sugar. I don't know what happened there. Something went pear-shaped there. Each contains Fe2+, and then each of those can bind with oxygen. So one hemoglobin, each contains Fe2+, so one hemoglobin molecule, can pick up 4 oxygen molecules. Now, I don't know if you need to know the reaction, but the reaction pick up 4 oxygen molecules, yeah. So each, what the hell is wrong with this thing? Yeah. That's the oxyhemoglobin. There's actually a reaction you can write for it if you wanted to know. There's a reaction as well. How does the reaction go? That'd be lovely if I knew, if I could remember. So you've got hemoglobin plus O2, and then you've got to balance it. Of course, it's reversible, the old chemical, back to your days in chemistry. And then that forms oxyhemoglobin, which is like, what's the formula for oxyhemoglobin? It's going to be 8 oxygens. We said 4 hemoglobin can pick up 4 O2, and then they form oxyhemoglobin. And I'll quickly pretend, because it's late, my brain's fried, I'll quickly pretend that I can remember the formula of oxyhemoglobin. So yeah, okay, it's just HbO2/4. Coolio. All right. I don't need 4. Why have I got 4? I don't know why I decided to put a 4 there. You don't need a 4 there. Just Hb plus 4O2. Yeah, because it's got 4 Fe2 pluses in 1 Hb. So hemoglobin plus oxygen makes oxyhemoglobin. You've got a reversible symbol there. Cool. All right, nice. I'm going to do a live in the daytime tomorrow. As I said, you can always rewatch it. It'll be there recorded. Like after this live, this will be recorded for a while and just hanging around on the internet. If you want any topics you want to go over, just put them up in the chat. If there's any topics you want me to go through or any particular style of questions, I can go through. All right, nice one, guys. So you can just add them in the chat even after I've left now. I'm going to go off now. So have a good sleep, and you've got one more day to revise for the dreaded paper 3. Just make sure with the essays, you know your 4 or 5 topics, and then let your brain and adrenaline bring out the detail in the exam. All right, guys, take care, and leave on the chat any topics you'd like me to go over tomorrow. I might do one tomorrow evening, but I might do one in the daytime as well. So, all right, guys, take care. Coolio.